Every single cell in your body contains the same DNA. However, not all of your cells are the same. You have nerve cells, blood cells, skin cells, bone cells, and many more different types that all have a slightly different structure so they can carry out their function. So, how can the same DNA create a whole range of different cells? The answer lies with differentiation. You were once a fertilised egg, a single cell called a zygote, which became a ball of cells through cell division. These identical, unspecialised cells are known as embryonic stem cells. Embryonic stem cells have the ability to become any type of cell in the organism. You can find out more about them by watching this video. When you were about 8 cells big, the process of differentiation started. The embryonic stem cells began to specialise to form different types of cells, which form different tissues and organs. Differentiation happens because genes in the DNA are switched on or off, and the cells start making different proteins. The set of genes switched on in one type of cell, say a red blood cell, is different to the set of genes that are switched on in another type of cell, like a muscle cell. This combination of genes allows the cell to make different structures, which results in the cells looking and functioning differently. Stem cells do exist past the embryo. They are called adult stem cells. Adult is a little misleading, as these stem cells are actually found in infants and children as well as adults. Whereas embryo stem cells can turn into any cell in the human body, adult stem cells are only capable of forming a few different types of cell. They typically form the type of tissue in which they are found. Adult stem cells maintain the body's tissues and organs. They can divide and reproduce indefinitely. For example, stem cells inside your bones, in your bone marrow, can differentiate to become different types of blood cell, but only blood cells. This is important because without this process you would not be able to create more blood cells to replace the ones that become damaged. And you create lots, an amazing 2 million new red blood cells every second. So differentiation in adult humans is mainly used for repair and replacement. Unlike humans, adult plants retain the ability to use stem cells to form all types of specialised cells. Plant stem cells are found in regions called meristems, which are located in the tips of growing roots and shoots. They can differentiate to form any type of specialised plant cell, such as xylem or phloem, so plant stem cells always act like human embryonic stem cells. If you take single cells from meristem tissues, you can grow a whole new plant from it. This is the basis of tissue culture and is used for cloning plants. In fact, nearly all commercial bananas sold in supermarkets around the world are from cloned banana plants. So, to summarise, in this video we have discovered that genes are switched on and off to form differentiated cells. We've looked at how differentiation in human embryonic stem cells means all the different tissues and organs can be formed, and how adult stem cells can only differentiate into a few different types of cell. However, in plants, stem cells in meristem tissues can differentiate into all cell types throughout the life of the plant.